Aloha mai kākou, I'm Dr. Alika Maunakea, born and raised here in Waianae, Hawaii. As an epigenetic scientist and health disparities researcher at the University of Hawaii, I co-direct the testing unit of the Pacific Alliance Against COVID-19, funded by the National Institutes of Health. My molecular biology lab at the University of Hawaii at the John A. Burns School of Medicine worked with our partners here at the Waianae Comprehensive Health Center to establish the first molecular diagnostics lab in a federally qualified health center in the state of Hawaii. This is important because this new lab provides our community with immediate access to the most accurate and reliable tests for diseases such as COVID-19. This test uses a technology called PCR, or polymerase chain reaction, which is a fundamental tool in molecular biology. If you've ever gotten this test before or curious to learn how it's done, we're here today to show you an insider's look into the lab.
After finishing the drive-through event, the nasal swab samples get sent upstairs to me for RNA extraction, okay? So this tube may look familiar. As you can see in here, you can see the end of the nasal swab that was used on the drive-through, and it's put in this liquid here. Now this liquid is used to neutralize the virus and basically kill it, to make sure it doesn't infect me as the technician. Now we can't say for certain that it's always going to work every time, so it's important that we perform this part of the experiment in this room that's labeled a contamination room, as well as this machine here, which is called a biosafety cabinet, that filters out air to make sure I don't breathe it in, has this protective screen, and when I'm done, shines UV light. Okay? So what we're gonna do is we're gonna take just the RNA from these samples, because remember, when we swab your nose, there's all kinds of things in there, mucus, bacteria, other types of things, but we want just the RNA because the RNA contains the COVID sequences that we're potentially looking for. Okay? So in order to extract the RNA, I'm going to prepare these blocks to put into a machine we'll look at later called the Kingfisher. It's an automated system that extracts the RNA, and I just have to set things up with the proper ingredients, okay? So you can take a look at this here. This is the block that these samples will go in. Every single cell you see is an individual sample, so I have 19 wells for 19 samples, as well as the negative control. The negative control is a test run we do to make sure that I did not accidentally contaminate anything that would possibly give us false results. Okay? The other blocks here contain various washing materials, also with the same filling pattern as the sample block. Eventually, at the end of the machine protocol, the extracted RNA will end up just in here, which is the elution plate. Okay? And how exactly does the RNA get extracted? Well, we use a special combination of reagents that looks like this. It's brown, it's really gross, looks like maple syrup kind of. It's called a binding mix, which contains magnetic binding beads, okay? And what these beads do is they attract the RNA to stick to the bead. And eventually, the bead will connect to a magnet inside the machine. The elution buffer will then take the RNA off the beads so that we just have RNA at the end, okay? So these beads are meant to stick to the RNA to pull it out, and it doesn't attract anything else in there, okay? So we can get started right now. I'm gonna use this extra long serological pipette. I don't wanna use this one because the tubes are very long and I could possibly cross-contaminate samples, okay? So it's very important that I use this long one, and I use one tube per sample and throw it away every time. Okay, here's sample number 1. Okay, so we're going to start with the first sample. Again, this is the original tube that is from the drive-through that may look familiar to you. And if you look in here, you can see the tip of the nasal swab that was actually put in your nose. So what I'm going to need is exactly 400 microliters of this sample. I'm going to pull some up and push it down again just to make sure everything is mixed. Going to pull up 400 microliters. I'm going to transfer it to the plate, and I'm going to repeat this process until all the tube contents have their own cell inside the block. And it's very important that I throw this tip away now that I've used it, because if I use it on another tube, I'm going to cross-contaminate and possibly end up with a false positive or false negative. This machine is called the Kingfisher. What it does is it extracts RNA from the nasal swab samples. So what this machine does is it contains magnetic beads inside the solution. Those magnetic beads get attracted to metal prongs in here. Those magnetic beads are attracted to the RNA, so it pulls the RNA out of the solution and leaves everything else that we don't want in there. Okay? So I'm going to load in this disposable tip comb tray. It's essentially there to make sure that the metal itself does not get contaminated. So I'll go ahead and place this in here. Next, it's asking me for the elution plate, which contains an elution solution. This is where the final RNA will be stored, and this is where we'll pick up with our process after this machine is done. This machine will wash over the magnetic beads that contain the RNA, and it's going to wash the RNA off of it, so we just get it in a solution. Next, we're going to add in 3 wash plates that contain various wash buffers and ethanol. This is to make sure, again, that we don't get any mucus, any parts of nose hairs, any bacteria, anything else that could possibly be in the sample. Okay, so here's number 3. And again, the nasal swab samples have been put into here with the magnetic bead solution, which works with the magnets as explained. Okay, so this machine takes about half an hour to run. As you can see, it sort of has an eccentric motion to it. After completing the RNA extraction, we're ready to test whether or not patients actually have COVID-19. Okay? So this is an RT-PCR machine. RT stands for reverse transcription, okay? Which means because COVID-19 is an RNA, we want to convert it back into DNA and then test to see whether or not patients actually have it or not. Okay? So this is a big machine here. I'm going to just hit open on the tray. We're going to place our completed plate into the machine to be read. Okay? So what we're doing is we're taking just the RNA that we extracted previously and we're looking at its sequence to see if it has certain sequences that are specific to COVID-19. And we use 3 different sequences in here, the N gene, the S gene, and the ORF1. Okay? And the reason we use 3 genes instead of 1 is that we want to make sure we have specifically COVID-19 and not any other coronavirus. So the usage of the 3 genes, the analogy I'll use is, let's pretend that instead of trying to find COVID-19, we're trying to find a specific person, okay? So let's say that the person's first and last name is John Smith. There's a lot of John Smiths in the world, right? But if we also include that person's middle name, they're probably the only John Smith with that middle name. So using 3 names instead of 2, we can make sure we're looking at specifically the right person. So same thing here. We want to have 3 different genetic targets so we know exactly that we're talking about COVID-19 and not any other disease. So we can take a look here. This is called an amplification plot. What the PCR machine is doing is that it's taking those specific sequences and it's making copies of them, right? So if the copies are being made, that's proof that the sequences actually do exist within the sample. And if the sequences exist within the sample, that means that the person does have the virus. So as we can see here, you can think of the x-axis as time, and you can think of the y-axis as the amount of amplification, okay? So we have, again, like I mentioned, the 3 genes. Take a look at the N gene. We see we have some amplification. If it's going up, that means, once again, the person most likely has the virus, because if there was no virus, they wouldn't have the sequence and nothing would be happening. So we take a look at the ORF gene, a lot of activity here. Look at the S gene, a lot of activity here, especially above this threshold, okay? And there are a lot of quality control measures that are put into this protocol to make sure that things are being correct and we avoid false positives and false negatives. One of which is the MS2 gene, or the MS2 phage rather, which is an internal control that makes sure that the machine is working properly and that I, as a technician, have not done anything wrong. I cannot say objectively by looking at these charts, I cannot classify a person as positive or negative, okay? So how we do an objective classification is that I'm going to take this file, place it into another computer that has an interpretive software that'll report objectively whether a person is positive, negative, or inconclusive, and also whether if I, as a technician, have made any mistakes. And this does a calculation on the file from the other computer, which will tell us objectively whether someone is positive or negative, and whether or not I, as a technician, made a mistake. Okay, so we can take a look here. Here's a run from this week. We have sample IDs here. Every test is ran in duplicate, so we can double check each sample to make sure that they agree, and yes, in fact, they're a positive, yes, in fact, they're negative, okay? Here we have the status. Valid means that I, as a technician, have done things correctly. Next, the interpretive result, okay? So as we can see for this same sample ID, A and B, which is the same person, run the test in duplicate. SARS-CoV-2 not detected, okay? Not detected for most of them, but as we come over here, we have 2 positive cases here. Come down over here, we have positive cases, okay? So after this, I hit export and it goes up to the server and they are contacted after that to begin quarantining, to begin any other preventative measure. So that's why I'm very happy to be a part of this effort here to provide a service in the community. And hopefully we can nip new positive cases at the bud as quickly as possible to prevent the spread here in our community.